Okay, so chapter 7, we're talking about cellular respiration, where we are getting energy from chemicals, specifically from food. So, is this a process of turning food into energy? Chewing on the leaves. Okay, so our formula or our equation for respiration, sugar and oxygen react to make carbon dioxide, water, and energy. Hopefully a lot of energy. Okay? And the energy is released from the chemical bonds in the sugars or in the starches. Okay. So the process of releasing energy from food is a good basic definition of cellular respiration. The energy is stored in the chemical bonds in the food. And ATP is the usable energy for cells to use for their work. Okay, so here we go. We have an uncontrolled reaction. Not going to help us much, going to explode the cells. Here we have using cellular respiration. Okay, we're going to take a little bit of energy at a time and collect that. So it's going to be controlled. And so we're going to end up with the same result of reaction, but we aren't going to explode the cell in the process. Okay, so this chapter is talking about the reaction for cellular respiration. Where does it happen? What do you need for it? What does it make? Okay, some things we need to know, some chemistry stuff we need to know. Oxidation is when you lose electrons, when you become positive. You also lose energy. You lose hydrogen atoms from carbon. Reduction, you gain electrons. You gain energy. You gain hydrogen from the carbons. Okay, so they're reverse of each other. And yeah, you need to make sure that we are not using reduction in lay terms, meaning use reduction to say getting smaller here. Okay, so redox reaction here. So our methane becomes oxidated. It gains, or it loses hydrogen. Okay, and our oxygen becomes reduced. It gains hydrogen. These reactions usually are paired together. So we need to look for these reactions being paired together as we study this. And they will be done by something called phosphorylation, which is adding a phosphate group to a molecule, like to change ADP into ATP. Okay, and that phosphate group, that bond there will hold the extra energy. Okay, so phosphorylation, we've got a chemical here. It has 3 carbons. It also has a phosphate. This enzyme is holding it and a partially charged energy molecule called ADP. It will allow the phosphate to leave the PEP and join to the ADP. That gives it 3, so now it's ATP. Now it is a fully charged energy molecule. And we have a molecule here called pyruvate, which we will become familiar with because it is part of cellular respiration. Okay, so the parts to cellular respiration, there's glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis, short, sweet process, get a little bit of energy. Krebs cycle, you get more energy. electron transport chain, get a ton of energy. So glycolysis, glyco is glucose, lysis is splitting, so we're breaking down or splitting glucose in half. This is found in all respiration. Doesn't matter if we are talking aerobic, anaerobic, fermentation, whatever. This was probably the first cellular respiration. This was probably the first way we got energy out of glucose. And its job is to split glucose, and you produce ATP, and you also produce another chemical called NADH. It happens in the cytoplasm, happens in bacteria as well as in eukaryotic cells, so it would have to happen in the cytoplasm. So glycolysis, we take glucose, we break it in half, that makes pyruvate, and we use that energy to make ATP. We also use it to make NADH. Okay, we have to think about the electrons and the electron carrier compounds. These are molecules that will transport or shuttle electrons within the cell. So they're going to move the electrons from one stage to the next stage. Okay, and so they can either be oxidized or reduced. Okay, so NAD. It can be NAD plus. If you give some electrons to it, now you have NADH. If you have NADH and you break electrons away, now you've got NAD plus. Okay, oxidized form, reduced form. Okay, and here we have diagrams showing what's going on there. We can reduce it, we can oxidize it. So we take and add the hydrogens to it, or we can break the hydrogens away. So what do we need for glycolysis? First off, we need glucose, obviously. We need some ATP. We need something to start the process. Need activation energy. We need some ADP so we can make ATP. We need some NAD plus so we can make NADH. What do we get out of it? We get 2 pyruvic acids. Each one has each one is a 3-carbon chain. Glucose has 6 carbons. We'll break it in half. 3 carbons and 3 carbons. You've got 2 ADP because remember we started with 2 ATP. We have to use their energy so they become ADP. We got 4 ATP. Remember we started with 4 ADP. When we used those 2 ATP and broke off and used that energy, we made 2 more ATP. And we took those 2 NAD plus and we made 2 NADHs. So they're going to carry some energy for us. So what do we get? For every glucose molecule, we get 2 ATP. That's our net results. We get 2 NADH. Net results. Okay, so we have to put some energy in, but we get more energy out than we put in. Second part is the Krebs cycle. Krebs cycle, also sometimes called the citric acid cycle or the tricarbolic acid cycle. Okay, it happens in the mitochondria. And we are going to oxidize pyruvic acid into carbon dioxide. And it is a cycle, so it takes several times through to get all the way done. It produces NADH and it produces FADH too, which this is another electron carrier. happens in the matrix of the mitochondria. So it happens in this very center part. Doesn't that look like a cool swimming pool? Sorry, got distracted. Okay, what do we need for the Krebs cycle? We need pyruvic acid. We get that from glycolysis. We need coenzyme A. That is, you find that in your mitochondria. We need some NAD+. We need some ADP. We need some FAD. Those are things that will take the hydrogens or the phosphates. And for every glucose we break, we're going to have 2 of each of these. We're going to have 2, 2, 8, 2, 2. What do we get in the end? We get 3 carbon dioxides. We get acetyl-CoA, okay? We get some NADH, we get some ATP, we get some FADH2. Those are all carrying energy. Get double that for each glucose. Okay, so we've got our pyruvate or pyruvic acid. There we have an NADH. There we have a carbon dioxide. We go through the Krebs cycle. Each time we go through it, we are breaking off carbon dioxide. We are getting NADH. We are getting ATP, and we're getting FADH2. So each time we're cycling through, we're getting some of this stuff. Okay? All right, and so the Krebs cycle is where we get most of our energy from NADH and FADH2. We get those energies, not so much the ATP as the other energies. We don't need oxygen. Didn't need oxygen for glycolysis. We don't need oxygen for Krebs cycle. So when we get these ATPs, they are from substrate level phosphorylation, okay? The inorganic phosphate is transferred from a substrate to an ADP, which then makes it ATP. So we're talking about something else, some phosphate joining in an enzyme to be paired with the ADP. Oh, and we're going to stop there before we get to electron transport chain. I will see you all later with this.